Welcome to our exploration of asexual reproduction, a fascinating biological process. Asexual reproduction is a biological process where a single organism produces offspring without gamete fusion or genetic exchange. This process results in offspring that are genetically identical to the parent. They are clones. This is fundamentally different from sexual reproduction, which involves two parents contributing genetic material. In sexual reproduction, genetic recombination creates offspring with unique genetic makeups, unlike the clones produced through asexual reproduction. To summarize, asexual reproduction is a process where a single organism produces offspring that are genetically identical clones, without the genetic variation introduced through sexual reproduction. The first key characteristic of asexual reproduction is that it requires only one parent organism. Unlike sexual reproduction, a single organism can produce offspring without the need for a mate. The parent organism undergoes various processes to produce new individuals. The second characteristic is that offspring are genetically identical to the parent. Since only one organism contributes genetic material, the offspring are essentially clones with the same DNA as the parent. The third characteristic is that asexual reproduction is generally faster than sexual reproduction. Without the need to find a mate or produce specialized sex cells, asexual organisms can reproduce more rapidly. The fourth characteristic is that asexual reproduction is common in simpler organisms. It frequently occurs in bacteria, many plants, and simpler animals that may not have evolved complex reproductive systems. Asexual reproduction may be triggered by specific environmental conditions, allowing organisms to rapidly colonize stable environments or respond to stress. The fifth key characteristic relates to the evolutionary significance of asexual reproduction. Asexual reproduction shapes the evolutionary trajectory of species through both its advantages and limitations. While it allows for rapid colonization and preservation of successful traits, the lack of genetic recombination limits adaptation to changing environments. These key characteristics make asexual reproduction an important reproductive strategy in the natural world. Binary fission is the primary method of reproduction in prokaryotes such as bacteria. In this process, a single bacterial cell divides into two identical daughter cells. The process begins as the bacterial cell elongates, increasing in size. Next, the bacterial DNA replicates, creating two identical copies. The cell membrane then grows inward, creating a division septum. Finally, the cell completes division, resulting in two identical daughter cells. Let's look at E. coli as a specific example. Under optimal conditions, these bacteria can divide every 20 minutes. This rapid reproduction leads to exponential population growth. After just 1 hour, a single bacterium can give rise to 8 cells. Budding is an asexual reproduction method where a new organism develops from an outgrowth or bud on the parent body. In this process, a small outgrowth or bud forms on the parent organism, develops into a new individual, and eventually detaches to live independently. Let's examine budding in unicellular organisms, specifically in yeast cells. First, a small bud forms on the surface of the parent yeast cell. As the bud grows, it receives a copy of nuclear material from the parent cell. Finally, when the bud reaches a sufficient size, it separates from the parent cell as a new individual yeast cell. Now let's look at budding in a multicellular organism, the hydra. Hydra budding begins with a small outgrowth forming on the side of the parent body. As the bud grows, it develops its own tentacles and mouth, becoming a miniature version of the parent. Finally, the fully formed bud detaches from the parent hydra and begins its independent life. To summarize the key points about budding, it produces genetically identical offspring or clones. It occurs in both unicellular organisms like yeast and multicellular organisms like hydra. Budding is an energy-efficient reproduction method for the parent, and it can occur repeatedly throughout the parent organism's life. Fragmentation is an asexual reproduction method where an organism breaks into parts. Each fragment can then develop into a complete individual organism. A well-known example is the starfish, When a predator bites off an arm, that severed arm can regenerate into a complete new starfish. The arm contains enough genetic material and specialized cells to form an entirely new individual. Planaria, or flatworms, demonstrate an even more remarkable ability. They can be cut into multiple pieces, and each piece will regenerate into a complete new worm. Scientists have shown that planaria can regenerate from fragments as small as 1/300th of the original worm. Each fragment contains special cells called neoblasts, which act like stem cells to rebuild the entire body. It's important to distinguish fragmentation from regeneration. While they may seem similar, they serve different purposes. Fragmentation is a reproductive process that creates entirely new organisms from fragments. Each fragment becomes an independent individual. In contrast, regeneration is a survival mechanism where an organism repairs or replaces damaged parts while maintaining its identity as a single individual. Fragmentation provides unique evolutionary advantages, especially in environments where rapid reproduction and habitat expansion are beneficial. Regeneration is a fascinating form of asexual reproduction where organisms can regrow damaged parts. In some remarkable organisms, this ability extends beyond simple repair to creating entirely new individuals from small fragments. Not all regeneration leads to new individuals. Let's compare the types of regeneration found in different organisms. The hydra exemplifies complete regeneration. When cut into pieces, each fragment can develop into a new, fully functional organism. Planaria, or flatworms, show even more impressive regeneration. They can be cut into multiple pieces, with each fragment developing into a complete new organism. While many organisms can regenerate parts, only some have the remarkable ability to produce entirely new individuals through regeneration. Complete regeneration represents a powerful survival mechanism that allows certain species to reproduce asexually from even the smallest fragments. Plants have evolved various methods of vegetative reproduction, including specialized stems that grow horizontally. Rhizomes and stolons are two such structures that allow plants to spread asexually, creating new individuals that are genetically identical to the parent plant. Rhizomes are horizontal underground stems that grow beneath the soil surface. They produce roots below and shoots above from nodes along their length, allowing the plant to spread laterally. Rhizomes serve multiple functions. They help plants propagate vegetatively, store nutrients, and survive harsh conditions like winter or drought. Common examples of plants with rhizomes include ginger, iris, and bamboo, which can spread rapidly through this method. Stolons, also called runners, differ from rhizomes in that they grow horizontally above the ground. A stolon extends from the parent plant along the soil surface until it reaches a suitable spot to establish a new plant. At the tip or nodes of the stolon, a new plant forms, developing its own roots and shoots while remaining connected to the parent plant. Stolons allow plants to efficiently colonize new areas by creating daughter plants that eventually become independent. The most familiar examples include strawberry plants, spider plants, and many grasses like Bermuda grass. Both rhizomes and stolons are efficient means of asexual reproduction, allowing plants to spread vegetatively without seeds. This strategy helps plants rapidly colonize favorable habitats and is especially advantageous in environments where flowering and seed production might be challenging. Sporogenesis is a form of asexual reproduction that occurs through spores. Spores are single cells that can develop into new organisms without fusion with another cell. A spore contains all the genetic material needed to grow into a complete organism. In fungi, spores are produced in specialized structures. These spores are released and dispersed by wind or water to grow into new fungi. Algae also reproduce through spores. Inside algae cells, spores form and are eventually released to develop into new algae. While spores are haploid in many organisms that use both sexual and asexual reproduction, asexual spores contain the full genetic material of the parent organism. This allows the spore to develop into a genetically identical copy of its parent. Sporogenesis is a remarkably efficient means of reproduction found across fungi, algae, and some plant species. Gemulation is a specialized form of asexual reproduction found primarily in freshwater sponges. During gemulation, sponges produce internal buds called gemmules, which are collections of archaeocyte cells surrounded by protective coatings. Gemmules are a remarkable survival mechanism that allows sponges to endure extremely harsh conditions. The tough protective coating contains spongin fibers and siliceous spicules that shield the internal cells from freezing temperatures, drying out, oxygen depletion, and predation. When environmental conditions improve, such as after winter or drought, the gemmules begin to develop into new sponges. The development starts with a dormant gemmule. When conditions are right, cells begin to emerge from the gemmule. These cells differentiate and organize to form a young sponge, which eventually grows into an adult sponge capable of producing its own gemmules. This reproductive strategy allows sponges to survive seasonal changes, disperse to new habitats, and rapidly reproduce when environmental conditions become favorable. Gemulation demonstrates the remarkable adaptations that have evolved in sponges, enabling these simple animals to persist in challenging environments for millions of years. Parthenogenesis is a form of asexual reproduction where an embryo develops from an unfertilized egg. Unlike sexual reproduction, parthenogenesis doesn't require fertilization by sperm. The offspring that develop from unfertilized eggs are typically genetic clones of the mother. In nature, parthenogenesis occurs in various animal species, including certain insects, fish, amphibians, and reptiles. Many types of animals can reproduce through parthenogenesis. This includes various insects like aphids and some wasps, reptiles like certain lizards, and even some fish species, including certain sharks. A fascinating example is the New Mexico whiptail lizard, an all-female species with no males. This species reproduces exclusively through parthenogenesis. The offspring of these lizards are genetic clones of their mothers, inheriting an identical genetic makeup. This species evolved through hybridization between two related lizard species. When a female reproduces through parthenogenesis, her eggs develop without fertilization. This means all offspring are genetically identical to their mother. Parthenogenesis offers several evolutionary advantages. It allows rapid reproduction without the need to find mates. It enables a single individual to colonize new habitats and preserve successful genetic combinations. However, there are also limitations. These include reduced genetic diversity, limited ability to adapt to changing environments, and potentially higher susceptibility to diseases. To summarize, parthenogenesis is a fascinating reproductive strategy where offspring develop from unfertilized eggs and are genetic clones of their mother. This process occurs naturally in many species and represents an important evolutionary adaptation. This reproduction method showcases the remarkable diversity of strategies that have evolved in nature. Rapid population growth is one of the most significant advantages of asexual reproduction. In asexual reproduction, a single organism can produce multiple offspring without needing to find a mate. This allows populations to grow exponentially, doubling with each generation. There are several reasons why asexual reproduction leads to faster population growth. Bacteria demonstrate this advantage perfectly. A single bacterial cell can divide every 20 minutes under ideal conditions. This allows bacterial colonies to form rapidly, with a single cell potentially producing billions of offspring in just 24 hours. Plants that reproduce asexually through runners or rhizomes also show rapid population growth. A single plant can produce multiple new plants, spreading quickly across available habitat. This growth pattern is exponential rather than linear, allowing much faster population increases than sexually reproducing species. This rapid population growth provides several competitive advantages, particularly in stable environments. Asexually reproducing organisms can rapidly colonize available habitats, maximize resource utilization, and outcompete slower reproducing species. Asexual reproduction offers significant advantages in terms of energy and time efficiency. Let's first examine how asexual reproduction conserves energy. Unlike sexual reproduction, asexual reproduction requires significantly less energy. There's no need to produce elaborate reproductive structures like flowers or specialized gametes. Organisms reproducing asexually don't need to spend energy searching for mates or engaging in courtship behaviors, which can be extremely energy intensive in sexual species. Time efficiency is another major advantage of asexual reproduction. Asexual organisms can reproduce whenever environmental conditions are favorable without waiting to find a suitable mate. Compare the simplified timeline of asexual reproduction with the more complex process of sexual reproduction. This time efficiency contributes to faster population growth and allows asexual organisms to rapidly colonize new environments when conditions are suitable. Asexual reproduction offers a significant advantage by preserving beneficial traits across generations. Unlike sexual reproduction, asexual reproduction creates offspring with genetic material identical to the parent. This perfect genetic copying ensures that advantageous traits are preserved without dilution or recombination. Beneficial adaptations such as drought resistance, high yield, and disease resistance are passed unchanged to the next generation. In agriculture, this genetic consistency is particularly valuable. Many commercially important crops are propagated asexually to maintain their desirable characteristics. Potatoes are propagated through tubers to preserve their starch content and disease resistance. Commercial bananas are genetically identical clones, ensuring consistent sweetness and seedless fruits. Grapevines are propagated through cuttings to maintain the exact flavor profiles needed for specific wines. This genetic consistency is particularly advantageous in stable environments. When conditions remain constant, maintaining well-adapted traits is more beneficial than generating genetic diversity. Since all offspring are genetically identical, there are no experimental combinations that might be less suited to the environment. Energy can be directed toward growth and reproduction rather than adapting new traits, allowing for rapid population expansion with proven genetic advantages. Asexual reproduction provides significant advantages for organisms in harsh environments. In extreme environments, finding mates can be difficult or impossible, making asexual reproduction a crucial survival strategy. These adaptations are vital in extreme habitats like deserts, Arctic regions, deep-sea environments, acidic waters, and high altitudes. In these environments, organisms face multiple challenges. Finding mates is difficult, resources are limited, and they must cope with extreme temperature fluctuations and water scarcity. Plants have evolved specialized adaptations for harsh environments. Bulbs store nutrients and water, allowing plants to remain dormant during unfavorable conditions. When conditions improve, these plants can reproduce rapidly without needing pollinators or other plants nearby. Animals also employ asexual strategies in harsh environments. Sponges form gemmules, which are dense cell clusters with protective coatings. These gemmules can survive freezing, drying, and oxygen deprivation, allowing sponges to regenerate when conditions improve. The timing of asexual reproduction is critical for survival. Organisms remain dormant during harsh periods, then rapidly reproduce when conditions improve. This enables survival and rapid population growth in environments where sexual reproduction would be challenging or impossible. Through these adaptations, organisms can thrive in environments that would otherwise be uninhabitable. Many organisms in nature cannot move to find mates. These immobile or sessile organisms face a unique challenge: how to reproduce without the ability to move. Plants are the most common example of immobile organisms. Trees, grasses, and flowering plants all remain rooted in one place. In marine environments, organisms like corals, sea anemones, sponges, and barnacles are fixed to the seafloor. Asexual reproduction solves this mobility problem by allowing organisms to reproduce without finding a mate. Through mechanisms like vegetative propagation in plants and fragmentation in marine invertebrates, these organisms can reproduce and spread without mobility. This allows them to efficiently colonize surrounding areas, maximizing their reproductive success despite their immobility. Asexual reproduction provides a key advantage in agriculture and horticulture: the ability to efficiently transfer engineered or selectively bred traits. This offers numerous benefits, including consistent crop quality, disease resistance, enhanced yields, and improved aesthetic features. Several specialized techniques are used to propagate plants with engineered traits. Let's look at three key methods: grafting, cutting, and tissue culture. Grafting involves joining the tissue of one plant with desired traits to the root system of another plant. This allows us to combine the engineered features of the scion with the established root system of the rootstock. Cutting involves taking a piece of a parent plant with desired traits and encouraging it to develop roots. This creates a new plant that is genetically identical to the parent, preserving all engineered characteristics. Tissue culture is a more advanced technique that involves growing plant cells in a sterile laboratory environment. A single sample can produce thousands of genetically identical plants, making it ideal for mass production of plants with valuable engineered traits. These propagation techniques have wide-ranging applications across different sectors. In farming, they ensure crop uniformity and predictable yields. For landscaping, they provide ornamental consistency and precise design control. In conservation efforts, they help preserve rare species and restore ecosystems. Ultimately, asexual reproduction methods enable efficient and rapid multiplication of plants with engineered traits, supporting sustainable agriculture, beautiful landscapes, and effective ecosystem management. In asexual reproduction, one significant disadvantage is the guaranteed transmission of harmful genetic traits. Unlike sexual reproduction, which allows genetic recombination that might eliminate harmful traits, asexual reproduction creates exact genetic copies. This means that any harmful mutation in the parent organism will inevitably be passed to all offspring, becoming locked in the lineage. This phenomenon is known as Muller's ratchet, named after the geneticist Hermann Joseph Muller. It describes how, in the absence of recombination, deleterious mutations accumulate irreversibly over generations, like a ratchet that only turns one way. Over successive generations, harmful mutations accumulate in the lineage. With no genetic recombination to potentially remove them, each new generation inherits all previous mutations plus any new ones. This has serious implications for asexually reproducing organisms. It leads to decreased fitness, reduced adaptability to changing environments, and can potentially result in population collapse over time. Unlike sexual reproduction, asexual organisms have no natural mechanism to purge these harmful mutations from their gene pool. Asexually reproducing organisms face a significant evolutionary challenge due to their genetic uniformity. When organisms reproduce asexually, all offspring are genetically identical to the parent, creating populations with little to no genetic diversity. When the environment changes, asexually reproducing populations struggle to adapt quickly. For example, if the temperature increases, all individuals with identical genes will be affected in the same way. Without genetic diversity, they may all struggle or perish if the change exceeds their tolerance range. In asexual reproduction, new beneficial traits can only arise through random mutations. In contrast, sexual reproduction combines genetic recombination with mutation, creating far more genetic combinations in each generation. The timeline for evolutionary adaptation is much longer for asexually reproducing organisms. Sexual populations can adapt in fewer generations through the rapid creation of new genetic combinations. While asexual populations must wait for beneficial mutations to occur randomly, which is a much slower process. This slower adaptation rate can be a significant disadvantage in rapidly changing environments, potentially leading to extinction if changes occur too quickly. In asexual reproduction, the offspring are genetic clones of the parent. This creates a unique disadvantage: genetically identical offspring that compete directly with their parents. Since these identical organisms share the exact same resource requirements, they compete for the same limited resources in their environment. In limited environments, this competition becomes more intense as the population grows. This increased competition has several negative impacts on the population. It limits overall population growth, restricts geographic spread, increases mortality, and reduces individual fitness. We can observe this competitive disadvantage in nature. For example, bacterial colonies often experience resource depletion at their centers, where cells compete intensely with their own offspring. Similarly, plant clones that spread through asexual reproduction may compete with each other for sunlight, water, and soil nutrients. To summarize, increased competition is a significant disadvantage of asexual reproduction. Genetically identical offspring have identical resource requirements, leading to direct competition with parents. This competition intensifies with population density, especially in resource-limited environments. Asexual reproduction has several disadvantages, including shorter lifespans in offspring. When organisms reproduce asexually, their offspring often don't live as long as sexually produced offspring. Scientific studies provide evidence for this disadvantage. Clonal organisms like water fleas show reduced longevity, and even asexually propagated plants may deteriorate over successive generations. Let's visualize this difference. Sexual offspring generally live longer than their asexual counterparts. There are several biological mechanisms behind this phenomenon. One key factor is telomere shortening. Telomeres protect the ends of chromosomes, but they shorten with each cell division. Another important factor is epigenetics. In sexual reproduction, epigenetic marks are typically reset during fertilization. However, in asexual reproduction, these marks can accumulate over generations, potentially affecting lifespan. To summarize, asexually produced offspring often have shorter lifespans due to accumulating cellular damage, unchanged epigenetic factors, and faster telomere shortening, highlighting one of the significant disadvantages of asexual reproduction. In many fields, humans have developed sophisticated techniques that utilize the principles of asexual reproduction. Just as organisms in nature reproduce asexually to create genetically identical offspring, humans apply these same principles to agriculture, horticulture, and biotechnology. These applications fall into three main areas: agriculture, where we produce food crops; horticulture, focusing on ornamental plants; and biotechnology, which uses laboratory techniques to manipulate plant cells. Let's explore the key techniques humans have developed to harness asexual reproduction. Plant cloning creates genetically identical copies of plants with desirable traits. This technique enables large-scale production of high-quality crops and ornamental plants. Tissue culture involves growing plant cells in sterile laboratory conditions. Scientists can regenerate entire plants from just a few cells, making it valuable for both conservation and commercial applications. Grafting combines tissues from different plants. For example, disease-resistant rootstock can be joined with stems that produce high-quality fruits, creating plants with multiple beneficial traits. These techniques have numerous real-world applications that benefit society in multiple ways. In agriculture, asexual reproduction techniques produce disease-resistant crops, ensure uniform harvests, and develop varieties with higher yields. These improvements are crucial for global food security. For conservation efforts, these same techniques preserve rare plant species, rapidly multiply endangered populations, and maintain biodiversity through seed banks and tissue culture collections. Overall, applying asexual reproduction principles offers several important benefits. These techniques enable rapid multiplication of plants with desirable traits, producing thousands of identical specimens in a fraction of the time needed for traditional breeding. They also ensure consistent traits in all offspring, which is particularly important for commercial agriculture and horticulture, where product uniformity is essential. Perhaps most importantly, these methods help preserve rare genetic combinations that might otherwise be lost, supporting both conservation efforts and the development of improved crop varieties. By harnessing the power of asexual reproduction, humans have developed powerful tools that enhance our food production, conserve biodiversity, and create new plant varieties with precisely controlled traits. In our exploration of asexual reproduction, we've seen a fundamental evolutionary balance at work. Organisms face a key trade-off between asexual and sexual reproduction strategies. Asexual reproduction offers advantages like rapid population growth, energy efficiency, and preservation of beneficial traits, while sexual reproduction provides genetic diversity, greater adaptability, and evolutionary potential. Rather than being limited to just one strategy, many organisms have evolved the ability to use both methods. Depending on environmental conditions, certain species can switch between reproductive strategies. Aphids reproduce asexually during summer for rapid population growth, but switch to sexual reproduction in autumn. Dandelions use both seeds and vegetative reproduction. Water fleas reproduce asexually in favorable conditions, but sexually under stress. This remarkable diversity of reproductive strategies has evolved over billions of years. From binary fission in bacteria to the complex sexual reproduction in mammals, each strategy represents a solution to survival challenges. Ultimately, the diversity of reproductive strategies in nature reflects different solutions to the fundamental challenge all life faces. That challenge is passing genes to the next generation in a way that maximizes survival and reproductive success in a particular ecological niche. Whether through rapid cloning or genetic recombination, each strategy represents a different solution to life's primary directive, ensuring the continuity of genetic information across generations.